Hello students. Today I am going to explain you chapter 11 biotechnology principles and processes, that is part 1 class 12th subject biology based on NCERT. But before explaining, I would like to request that kindly like, share and subscribe our YouTube channel so that you may always get an update of new videos. In this part, first I have to explain about the biotechnology. What is biotechnology? Biotechnology actually deals with the techniques of using microorganisms, plants, or animal cells or their components or enzymes from organism to produce product and process useful to human beings. Microorganisms were first to produce some organic compounds like citric acid, and some microorganisms produce antibodies also. These antibodies are very helpful for the human beings to fight against the different types of diseases, so that in their processes natural capabilities of the organisms and cells are exploited. These activities are called old biotechnology. These are the traditional biotechnology which we have to produce antibodies and citric acid, some enzymes. But in the modern biotechnology, the production is based on genetic engineering. Like example, human insulin is also produced from transgenic E. coli. In this uh, modern biotechnology, we have to use the transfer of genes. We have to identify the useful genes, and we have to cultivate the product of these genes. A definition of biotechnology that given by EFB, that is European Federation of Biotechnology. The European Federation of Biotechnology said that biotechnology is the integrated use of biochemistry, microbiology, and engineering science in order to achieve technological applications of the capabilities of microorganisms, cultured cells, and parts thereof. So this is the combination of microbiology, biochemistry, and engineering sciences to produce different types of enzymes and different types of uh, antibodies and proteins desired with the help of desired genes. Now we have to discuss about the principles of biotechnology. There are basically two main techniques of modern biotechnology. That is, one is genetic engineering. What is genetic engineering? This technique is used to alter the nature of genetic material. Genetic material, all of you know that DNA and RNA, these are present in the cells, and all the living cells having DNA and RNA, so that these DNA and RNA to introduce into host organism and thus change the phenotype of the host organism. That is the main concept of the genetic engineering. The another one technique is a chemical engineering. It involves maintenance of sterile microbial contamination free conditions in chemical engineering processes to have growth only the desired microorganism in large quantities. For the manufacture of biotechnological products such as antibiotics, antibiotics are useful drugs that help us to fight against the different types of diseases. Vaccine is also helpful for protecting us from the different types of diseases. Enzymes, medicines, and hormones. So that genetic engineering is a kind of biotechnology which deals with the manipulation of genetic material by man in vitro, in vitros, inside the lab. Inside the lab, we have to do all these things. Two main discoveries of genetic engineering are, that is, presence of plasmid. What is plasmid? Plasmid is found in the bacteria, that is an extra chromosomal genetic material which can undergo replication along with the independent of chromosomal DNA. In bacteria there are two DNA. One is chromosomal DNA and the another one is a plasmid, that is a extra chromosomal circular DNA. And the restriction endonuclease is one of the familiar example that is also known as molecular seizure, which can break the DNA and specific sites also called molecular seizure. So that restriction endonuclease is also known as molecular seizure, which can break the DNA at the specific sites. So these techniques we have to use, or these are the principles of the biotechnology. Using these principles, we have to make several types of antibiotic, vaccine, enzymes, medicine, and hormones for the human welfare. The techniques of genetic engineering. In genetic engineering, it includes formation of recombinant DNA, use of gene cloning, gene transfer. First, we have to find out the desired genes which are useful for the human welfare, and through this gene we have to introduce into the vector. Plasmid, that I have already explained, plasmid is used as a vector. So that gene is introduced into the vector, and this after the combination of the foreign DNA with the plasmid, that is known as recombinant DNA, and this is transferred into the bacterial cell, that is making multiple copies of that particular desired gene, so that it permits to isolate and introduce only one or a set of desirable genes without introducing undesirable gene into the target organism. So that traditional hybridization procedure used in plants and animal breeding very often lead to inclusions and multiplication of undesirable genes along with the desired genes. But in this technique, we have to use the only desirable genes. We have to find the genes which are useful for the human beings or for the human welfare. So without introducing undesirable genes into the target organism, a piece of DNA is introduced into the alien organism would not be able to multiply itself. The DNA which have introduced into the cell, that is not having capable to multiply itself, but in the organism. But when it get incorporated into the genetic material of the recipient, it may multiply and be inherited along with the host DNA. There is a specific DNA sequence called the origin of replication. Thus, the alien DNA linked with the origin of replication can replicate and multiply itself in the host organism. It is also called cloning, that is gene cloning. So that there is a specific sequence, students. So there is a specific sequence. You have to clear the concept in this part. There is a specific sequence known as origin of replication, so that the alien DNA linked with the origin of replication can replicate and multiply itself in the host organism. It is also called cloning or the gene cloning. So in the construction of the first artificial recombinant DNA molecule, the first recombinant DNA was constructed by Stanley Cohen and Herbert Boyer. They cut the piece of DNA from a plasmid carrying antibiotic resistance gene. What type of gene carrying by the plasmid? That is antibiotic resistance genes in the bacterium. Salmonella typhimurium is a bacterial name. So cutting off a piece of DNA from the plasmid with the help of restriction enzyme. The piece of foreign DNA cut from the plasmid was linked with the plasmid DNA acting as a vector, which was done with the help of enzyme DNA ligase. DNA ligase, which is very helpful in the ligating the foreign DNA into the plasmid, which act on cut DNA molecules and join their ends. This is newly formed DNA having integrated fragment of antibiotic resistance gene is called recombinant DNA. This is called recombinant DNA, and the vector used to transfer recombinant DNA to E. coli. So this vector is used to transfer recombinant DNA into the E. coli bacteria. In presence of DNA polymerase enzyme, make multiple copies of recombinant DNA. So in this way, we have to make multiple copies of the recombinant DNA molecule. There are three basic concepts in creating genetically modified organism. First, we have to identify the DNA with desired genes. Gene is a part of DNA. That is also, gene is a small part of DNA. So first that we have to identify DNA with the desired genes. Introduction to the identified DNA into the host, and maintenance of introduced DNA in the host, and transfer of DNA to its progeny. So in this way, we have to use the making the recombinant DNA molecule. This is the topic of this chapter today, that is principles of biotechnology and techniques of genetic engineering. Some of the questions, like you have to write the full form of EFB. What is EFB? What are the principles of biotechnology? And what do you understand from recombinant DNA, gene cloning and gene transfer? Explain the role of plasmid. That I have already explained. In bacteria, there are some extra chromosomal genetic material known as plasmid. And plasmid is used as a vector. That is frequently used as a vector in the biotechnological process. Thank you, students. This is all about the topic. Today's topic, that is principles of biotechnology and the techniques of genetic engineering. Thank you, all of you.